Welcome, everyone. It's a pleasure for me to facilitate this session tonight. This is the tail end of a beautiful day of science here at McGill. I would like to start by thanking the Garner Foundation for making this meeting possible, as well as our sponsors, and McGill as well. Tonight's session is about essentially reaching out and with science, the latest science on RNA-based therapeutics. RNA-based therapeutics made a huge difference, and they captured the world's attention with messenger RNA vaccines that essentially led us out of a pretty dramatic pandemic. But what is often missed when you look at the potential of those therapies is that they can be used for multiple other purposes. We'll see together tonight some of those modalities, the potential disease that it can target and cure. Of course, a lot of it is still experimental and will require quite a bit of research and a hard look at the data, but it bears a lot of promise, and the fact that it moves very, very fast necessitates a good look at how they function, what's their potential implications, and essentially how they will end up changing what's happening in the clinic. So this is my role tonight, essentially as a facilitator with my starting introduction. And then we'll have a panel when I'll invite 4 panelists from different horizons, scientific horizons, to come in front. We have Mohammad Gabriel Alameh from Penn Institute for RNA Innovation. We have John Androsevich, Global Head for RNA Medicine Lead at Pfizer. We have Jan Joly to address some of the ethical questions and Director of the Center for Genomics and Policy at McGill University. And finally, we have Anastasia Khorova from the RNA Therapeutics Institute at UMass Chan Medical School. They cover different aspects of the expertise. And I hope we'll have a good session debating the potential of these therapies, as well as eventually how and when those modalities will end up impacting at the clinic. So at the end of my introduction, I'll invite you in front. Yeah. All right, I have a short intro. That's what I should start with. Okay, so my talk today will essentially start with the impact of those RNA-based therapies. A good example is the vaccines that essentially, based on excessive debt and credible sources, save up to 19 million lives worldwide in the first year of implementation. They're not all traceable to the Moderna and Pfizer vaccine, but those vaccines were actually progressively supplanting the other vaccines because they had fewer side effects, and they were ultimately, especially in Quebec, took the lead essentially in the applications. There's a rapid growth in terms of clinical trials for RNA-based therapeutics. We know now just only with the RNA vaccine modality that there's right now 3,400 new RNA vaccines in the works of all sorts of modalities. This is from clinicaltrials.gov source. So this is really accelerating, and about 100 of them are against cancer. And this is just the first flavor telling you that it's not all about viruses, and there's actually many other applications for those RNA-based therapeutics, and that can change people's life quite dramatically. The market for this, there's a lot of pressure now for this, and this is very speculative, but it's from credible source from Allied Market Research. And what you can see is that at the end of 2021, the market was for about $5 billion worldwide, and it projected with the other modalities and acceleration of RNA-based vaccines to reach about fivefold, $25 billion by 2023. So there's a potential there for the new modalities to expand, but also for RNA viruses to develop towards other targets, and that is not even considering, for example, possible new pandemics that may emerge. I want to take my hat here of university professor and talk a bit about the basics of RNA. It's the coolest of molecules, really. It's a polymer that's slightly different from DNA simply by one residue called at the position 2' hydroxyl. And instead of having hydrogen, you have a hydroxyl here, and this changes everything. It actually makes the molecule very unstable, prone to degradation, either enzymatic or chemically, but also RNA likes to behave a little bit more erratically by also forming structures that are double-stranded, a bit like DNA, but it doesn't have the same geometry, and it's much wilder in general. So there's structure, but it's not the same as DNA, and this allows RNA to carry out multiple functions, some of them even enzymatic. So it's something that people don't know. Before proteins, before the hard work of proteins, RNA was doing the bulk of the functions in your cells. We used to call it the RNA world, and there's still vestiges of this in your biology right now. Another difference between DNA and RNA is that RNA use uracil instead of thymidine. And for those of you who are chemistry aficionado, there's a lot of variation here, a modification that the cell uses to distinguish this nucleotide from another one, different RNA. So there's some cool chemistry on this residue in particular. RNA is a polymer, and one of the consequences of that polymer is that it has a phosphate backbone. Don't want to go too much into chemistry, but imagine heavily charged polymers. So large molecule with a very strong negative charge all along the backbone. And one consequence of this backbone being negatively charged is that it doesn't move really well across membranes. Your cell's membranes are hydrophobic, what we call hydrophobic. And anything with a charge or polar doesn't move across really easily. So this essentially is kind of daunting when you want to consider RNA as a potential treatment. Moving in cells, carrying out function, being translated into proteins, for example, is not easy with something like this. This is the bottom line of the function of RNA in the central dogma. So you have DNA being essentially the hard copy and the memory of the cell. You know, whether you have blue eyes or whether you will have, you're going to weight a certain weight or measure a certain height. RNA is the messenger RNA in particular is the intermediate between the DNA and the proteins. That's traditionally the view in the central dogma. So in many ways, RNA is like the messenger, and it's a perfect name for it. But there are actually multiple other functions for RNA. We'll talk a little bit about those later on. But that's the central dogma, and that's where the messenger RNA comes from. It's a messenger between DNA. It carries out the code that will ultimately lead to production of proteins. So if you take an analogy, for example, for a messenger RNA, you have the hard drive in your computer, and you need a temporary map for which you want to build essentially your house. That map is transient in nature, so you're not going to keep it forever. It's probably not as reliable as the hard drive that is the failsafe of what your species is and whether your cells is healthy or not. So this is kind of a transient nature, but you still need it to be mobile and carry out the information to build proteins, for example, enzymes, hormones, nails, or whatnot. And other things for other uses for it, for example, are antigens, things that will generate antibodies ultimately if you stimulate the right cells for an immune response. So one of the breakthroughs why at this point we're considering RNA therapeutics as really exciting and with a lot of potential is actually the discovery or the development of structures called lipid nanoparticles. You want to think about essentially as tiny bubbles made of fat, but they can be guided also. And essentially, the key here is that it protects the nucleic acids, the RNA that has this 2' hydroxyl and makes it very unstable, but also polar, and it hides it from the membrane. So it can be carried around, and modifying the chemistry of the surface of that bubble actually allows it to reach different compartment and has different distribution, what we call pharmacokinetics in drug language. And it was really the key because the barrier for RNA as therapeutic really was this instability, systemic instability. You'd get it in an organism, and it'd get degraded right away, and it would move across membranes and then get into organs really easily. It is biocompatible because it's made of building blocks, the lipids and derivatives, that is chemically quite close to the normal components of the cells. Actually, it mimics things that are really close to lipoprotein complexes that circulate in your blood, and it probably use some of the pathways that are similar. It can be modified, and that's really cool because you just change your recipe of the lipids in that bubble and you can lead it to a different organ. or change its behavior, make it stay a little bit longer, protect your RNA a little bit better. Another component here is that you have cationic lipids. Cationic means positively charged, and this has really good interactions with your RNA. So actually compacts it, we call it encapsulation, and these are the formulations essentially that led to the messenger RNA therapeutics, but also other small RNA-based therapeutics that are out there use these modalities. So this technology has a huge impact on, it essentially brought down a major barrier for the use of RNA therapeutics. And this happened in recent years through innovation, namely that was done in Canada in good part. Okay, so the other button. All right, so if you think about it at the simplest form, right? You want to use RNA as a drug, as a medication. Well, you formulate it, you hide it from essentially the environment, you protect it with a lipid nanoparticle, and now you have essentially a disease protein, and you want to replace it. So let's say this DNA, this gene here produces an RNA that's defective, that doesn't produce protein or makes a sick protein or is being degraded. What you can do is just take a messenger RNA that encodes the healthy messenger RNA, package it into one of the lipid nanoparticles and inject it in the organism. It's not always intramuscular. There's different ways or pathways to distribute it, but the result is the same. You're literally replacing a disease messenger RNA with something healthy. That's the simplest modality of RNA therapeutics you can think of. And there are some of those therapies are based like this. But the one that I just described earlier on is the use of messenger RNA as vaccines. So when you have a messenger RNA encapsulated in an LNP, for example, you can make the code inside this messenger RNA sequence so that it encodes an antigen that once injected into a patient or someone in the middle of a pandemic, for example, would protect it ultimately by producing an immune response. The mechanism is a bit complicated. Immune biology is really cool. In essence, what happens is that the RNA encodes for the antigen. The antigen is presented on the surface of the cell, and there's some immune cells that will see it and say, hey, that's not mine. It's probably a virus. I got to develop some antibodies against it, and that drives the immune response, ultimately, the antibodies that protect you from infection. So that's the messenger RNA as a vaccine modality. There are other flavors of RNA-based therapeutics. One of them is the use of RNA or DNA as antisense oligonucleotides. Different nucleic acids can be used as nucleotides. This has been around for a while. It's been quite successful for a very specific disease. And what's really cool about ASO is that very often it targets genes that are said, or diseases are meant to be said to be orphan, for which there is no other option, no easy one anyway. So this is quite versatile, and you can use it two ways. There are several ways to use it. One of them is essentially to guide it so that it binds to a messenger RNA, and it's recognized by an enzyme called RNase H that recognizes the hybrid and cuts the messenger RNA and degrades the messenger RNA. You would do that for a messenger RNA that for example, is responsible for disease, or if you know that knocking down or deleting this messenger RNA has an impact that's beneficial or therapeutic. Another cool use for these ASO, antisense oligonucleotides, is that you can change essentially constitution of messenger RNAs. I'm going to shortcut here, give you the cheat sheet, and the idea is that you can essentially change the combinations of so-called blocks that constitute the messenger RNA, so-called exons. And normally, RNAs are assembled by a process called splicing, and these ASOs actually can switch the use of these exons and connect some that are not normally connected. And some of the diseases are actually generated or a consequence of the use of wrong exons, and you can influence the combination of these exons using antisense oligonucleotides. There are other modalities, but I would say these are the two main ones that are used for antisense oligonucleotides. This one is really dear to my heart. It's a pathway called RNA interference, and essentially the use of small RNAs that are usually produced as heteroduplex that are loaded into a complex called Argonaut. And the goal of the job of this complex essentially is to find a messenger RNA. It just seeks messenger RNA inside the cell and cleaves the messenger RNA through an activity called slicing. So this is a process called RNA interference, and there's quite a few drugs that are coming through, and we see an acceleration of FDA approval improve drugs that are based on this mechanism. And ultimately, the goal is to degrade or silence messenger RNA. Again, it's the same thing as with the ASO. You want to essentially shut down a gene that you know is responsible for your disease, or if you shut down a disease when you know that it has a therapeutic impact on your organism. So in a nutshell, before our panel, we can replace a product of a disease gene. You can silence a messenger RNA. These were the siRNAs. You can alter the active form of messenger RNA using ASO, like these building blocks of messenger RNA can alter. And finally, you can harness the immune system through the generation of antigens, essentially the modality called the messenger RNA vaccine. There are others. I'm keeping it short. Some of them are pretty, have an ethical consequence, and there's consideration there for safety as well. One of them is CRISPR-based or genome engineering, which essentially use RNA as a guide to change the DNA sequence. We won't talk too much about those. We probably will have questions about those. But the most important thing about RNA therapeutics, the most important thing, is the versatility of its repurposing. So you can essentially redirect an RNA therapeutic just by changing the sequence of the RNA that is being used. So if you change the messenger RNA to a different sequence, you may have the same kind of bubble protecting it, but it will encode a different protein. It will actually ultimately lead to a different outcome for your health. So, and the same for siRNA. So you change the sequence, you may have a completely different target and a different outcome for health. So the versatility here is the key. Just called information therapeutics. So just by changing the sequence of those therapeutics, you can actually repurpose them. And when you think about it, this opens the door to every gene being either replaced or shut down or tuned for therapeutic purpose. So this is much beyond and much faster than what the traditional therapeutics development actually envisioned. So that's what we're facing now, this huge potential and this very accelerated onset and development of new therapeutics for multiple modalities. So with this, I'll invite the panelists and we can discuss all of the implications of these different modalities in therapeutics. I mentioned the panelists, and we'll have two blocks of questions. One of the series of questions are selected. It's going to be the first 25 minutes or so. They're questions that we've seen a lot in the polls and what people have asked. Then we'll go on for about 20-25 minutes again with questions that were asked by individuals. We'll name the individuals. And then if we have time after all of this, we received something like 46 questions so far. If we have more questions after that, we'll see and address them as well. So, you ready? Let's do it. Well, first I want to thank the panelists for being here, because we're researchers and we're quite comfortable in our labs. But what you see today are researchers actually stepping up to the plate and engaging the public. That's the purpose of this public series, this public forum. So I'll start with something that was left front and center in the questions from the public. What other diseases beside COVID can RNA therapeutics treat? And you just pick whichever, whenever your heart feels like it, just pick it up.
I can get started with that. This is what diseases currently or potentially?
Potentially.
Potentially. Many. And really, I think it speaks to the fundamental nature of RNA as a genetic medicine. It's the perfect complement to our current approach to understanding disease, to intervening in that disease, and that is through genetics. And so the ability to have a genetic tool, a genetic drug, in order to really take advantage of the genetic understanding of the disease can really provide, I think, breakthrough in a lot of different diseases. So more specifically, I think there's additional infectious diseases, vaccines, prophylactics that we'll be seeing, and certainly we saw the numbers for the clinical trials. That's going to be applied and is currently being evaluated for cancer and oncology. There's additional, a lot of emphasis on rare disease. Rare disease is a testing ground for a lot of these new RNA platforms because they're genetically defined a lot of the times. They're what's known as a monogenic disease. You have one gene gene that has a defect. You can use this sequence-based, this genetic-based modality, whether it be the ASOs, the siRNAs, or the messenger RNA, in order to provide a correction to that single gene defect. Once you have demonstrated that concept, you've de-risked your technology. That gives you confidence to move forward into larger, more complicated diseases. And I think we're going to start to see more of that into the future. Cardiovascular disease, diabetes, obesity, some of the biggest health crises that we're facing today, RNA could play a role in that. I do want to say, though, it's not just going to be RNA. It's not, we're only looking at a future of just RNA medicines. I think it's going to be a really powerful tool in our toolbox, another arrow in our quiver, if you will, and it's going to be quite effective, I think, for certain areas and certain diseases where some of these other modalities haven't necessarily been successful because the targets are known as undruggable targets. This gives you an orthogonal approach to actually intervene in these diseases.
All right, so this is John Androsovich from Pfizer, and thank you for picking the first question. I want to just mention one thing, I forgot to mention it. It's that you can ask questions in French. Ça va me faire plaisir de répondre à vos questions en français, de les inclure puis les traduire. Puis à chaque fois qu'il va y avoir une séance ou une réponse à une question clé comme celle-ci, je vais la traduire aussi en la résumant. Ça va faire plaisir de répondre à vos questions en français aussi, même si la présentation est en anglais. So,
Can I
yeah,
add
you can
a
go
little
ahead,
bit?
please.
So I think absolutely that initially we started with orphan indications and genetically defined diseases, but what have been seen in the last couple of years, it's transformation of the field where we went from populations of less than 10,000, sometimes it's 5,000, sometimes it's 500 patients, and sometimes it's even N01. But right now there is a clear trend towards transitioning towards major clinical indications which will have impact on how the medicine is being practiced pretty much for everyone. And I think there is a lot of considerations related to that because they are also related to economics of medical care and how the medicine is being practiced. And one of the things, right now there is a lot of discussions about the cost and the market size, but I think the majority of the cost of the drugs is related to the cost of the drug development. Because the manufacturing per se, maybe gene therapy excluded, cost very, very little. So when the drug becomes generic, and this is true for majority of RNA drug, is the cost of the drug substance itself is on par with the cost of the tube or syringe with which it's being injected. So right now, there are several drugs, at least in chemically modified siRNA field and with the COVID vaccines, which has a major potential, have a major impact on human cells. And the durability of the action, like with fully chemically modified GalNAc siRNAs, there's 4 drugs approved, and single injection gives you 6 to 12 months duration of effect. The one which is most likely a lot of people, even in this audience, which age might need to take, is the one which is lowering cholesterol, and its name is inclisiran. It was developed by the company of Alnylam, and I'm not associated with it in any way or form. But it's fascinating because it's a single injection once in the 6 months, which essentially gives you a very important reduction in level of cholesterol. And this is affecting not 500 patients, it's affecting wide range of patients. Another one which is under development is going after angiotensin, which is high blood pressure, yeah? High cholesterol, high blood pressure, aging population. So we are talking about millions, if not billions of patients affected by it. So I think there will be, with advancement of RNA medicine, both for immunization as well as for major clinical indications, the way the medicine is going to be practiced in the future, and I think the cost of medical support is going to change dramatically.
Yeah, just so I can summarize in French before I lose some information. John mentioned that some of the targets, en français maybe. So John Androsovich de Pfizer mentionnait que des maladies comme des maladies rares ou on les appelle orphelines par exemple, vont être accessibles aux thérapies de l'ARN. Il mentionnait d'autres maladies aussi chroniques comme par exemple le diabète pouvaient être traitées par ces nouvelles modalités. Il y avait beaucoup de potentiel pour ces thérapies-là, par exemple. Puis une autre chose que John a mentionné, c'était qu'il pourrait être utilisé en combinaison ou pour complémenter l'arsenal actuel de thérapie. Ça ne va pas nécessairement supplanter complètement les thérapies traditionnelles, mais ça va juste étendre. Il y a beaucoup de potentiel pour étendre finalement les outils qui peuvent être accessibles. Anastasia mentionnait quelque chose de très important, c'est-à-dire que le coût de ces médicaments-là va être affecté par la diversité et l'efficacité de ces traitements-là. Elle mentionnait l'exemple, par exemple, de modalité à l'ARN qui permet de diminuer le cholestérol dans le sang avec une seule injection sur des longues périodes, par exemple. Puis la production de ces médicaments-là est beaucoup moindre, par exemple, puis ça rapproche pas loin du prix finalement du tube au bout de la ligne. Donc le marché va changer, puis les options pour différents traitements vont changer au bout de la ligne. Puis Anastasia mentionnait vraiment l'immense potentiel de cette thérapie-là. You wanted to add, Mohamed Gabriel?
A couple of things to add, actually, since Anastasia mentioned actually the immunization. So when we're looking into mRNA vaccines per se, one thing that we always need to keep in mind is we're not changing the biology of the pathogens. So there's certain pathogens that are very difficult to raise a vaccine against, right? Like HIV remains HIV. It has very complex biology. So we have to use or be careful actually about saying that these mRNA vaccine will work for any pathogen, right? They would work. They have to be supplemented with other strategies as well. I've seen strategies where people are using mRNA LNP vaccines plus adjuvants with recombinant to create certain type of immune responses. But depending on the pathogen, depending if it's mucosal or non-mucosal, there's a lot of challenges. We're going to reach, I would say, a point where we would be able to control a lot of these pathogens, but biology remains biology, and we need to understand this first before developing anything else.
All right, thank you, Mohamed Gabriel. Are we ready for another one? Mohamed Gabriel mentionnait que certaines de ces modalités-là mobilisent essentiellement des fonctions biologiques, ou la fonction biologique du pathogène, par exemple. Donc, c'est vraiment quelque chose de très précis qui mobilise les connaissances précises qui sont dans ces voies-là. En résumé, c'est ça. Mais Mohamed Gabriel, tu peux complémenter, tu parles français aussi. OK. Oui. OK, let's move to a second one. You ready? When will it happen? When will we see those new modalities in the clinic? When will it actually change people, the way people are treated? We've seen already with the vaccine, but when is all of this wave of therapeutics going to happen?
I can say for diseases involving liver, it's happening now. This is not a fantasy. There are 4 approved drugs with GalNAc conjugates. There are several dozens in clinical development. It took almost 18 years to get the first one developed, and after it showed clinical proof of concept and efficacy and safety, then it's exponential acceleration. So for cholesterol, you can get it from your doctor today. I'm not sure how much it cost and how whether it's available in Canada or not, but it's reality today. And I think diseases beyond liver, I will say there will be approved drugs within 3-4 years from now.
I'll add to that and say, I think for certain rare diseases, I know for certain rare diseases, RNA has been transformational for the lives of patients. I had previously mentioned that rare disease, these monogenic diseases, is often a testing ground for the technology. It's not to diminish it. It's not just a means to an end. It is the end for certain patients. It is so transformational for their lives. And another one besides the liver has been the approval of Spinraza for spinal muscular atrophy. These young children who are no longer able to walk or sit up in their beds can do so after a couple of injections of Spinraza. It's amazing how this has really impacted the course of this disease. And it really comes down to understanding the pathology of that disease and then how you can quite intuitively design an RNA to actually correct that pathology. And I think that's fantastic. So by my count, I believe there's 18 approved RNA medicines or have been approved. Some of them have been discontinued. That 18 includes the 2 mRNA vaccines as well. And then I'll give it one bonus point for melicin, which is the N=1 disease for the Batten disorder for Mila. That was really an incredible effort. It was an exception, but perhaps it could be a signal of what could come. And I think that especially using that ASO technology that Thomas had introduced, it does give you the ability to have these highly personalized medicines. And I think we're seeing that, I know we're seeing that play out now. Now, part of the question was, when are we going to be able to be, when are we collectively, most individuals that fortunately are fortunate to not have these rare diseases. I think that is coming, I think, to Anastasia's point. I think that we're going to start to see this more rapidly. We already have the vaccines, obviously. Flu vaccines are in the works too. This could be a seasonal type of treatment with messenger RNA. And so whether you are going to the clinic and getting a vaccine, whether perhaps you are prescribed a subcutaneous injection that you could apply at home, or perhaps you would need to go to a clinic to have an infusion, I think that what we're going to start to see is that our neighbors and our family and eventually us are going to see RNA as another option that is provided to us by our physicians. It will be an option, and there will be other options, of course, too. But this is another approach to treat your disease.
All right. La question, c'était quand on commencera à voir des impacts de ces nouvelles modalités de thérapie à l'ARN. Anastasia a commencé par dire que c'est déjà en marche. Il y a déjà des exemples de thérapie, par exemple, qui sont disponibles. Il n'est pas trop sûr combien ça coûte pour l'instant, mais c'est disponible pour certaines maladies bien spécialisées. Puis c'est déjà en marche, ce changement-là. John a mentionné, par exemple, que dans son cas, il y a un cas de maladie rare où il a vraiment été frappé par l'impact de ces thérapies-là pour des gens qui n'avaient aucun autre traitement. On appelait ces maladies-là maladies orphelines. Puis pour ce qui est de quand ça allait progressivement ça allait arriver, ces changements-là, pour les types de thérapie, John mentionnait qu'il pense que progressivement à la clinique, il va y avoir de plus en plus d'offres de ces options-là basées sur l'ARN. Donc, ils vont vous offrir des thérapies, certaines traditionnelles, puis d'autres progressivement de plus en plus d'options fondées sur les thérapies à l'ARN.
I just wanted to add one thing resonating to what you said. Biology matters, and biology will define ability to develop cures. And right now, because of the overall excitement, we are a little bit, as a community, trying to build the plane while we're flying it. And it clearly did happen with mRNA vaccines, and I think we as a community as a whole did a fantastic job. But with that said, some mistakes will be unavoidably made, and this is like ethics of how we approach orphan indications and how we go about design of the clinical trials is absolutely essential. With N01 initiative like ultra-orphan indications, they have been some setbacks because we are also learning what works, what doesn't work. It's not an absolute golden bullet. It's not going to be applicable to every single thing, to a lot of them. But it's something we as a community have to kind of act responsibly, think about the ethics of it, not overpromise, let's say underpromise and overdeliver.
Oui. Anastasia fait des points très importants sur le fait que tous ces changements-là ont lieu très rapidement, puis que pour bien développer, bien capitaliser là-dessus, puis diminuer finalement l'impact potentiel des nouvelles thérapies, il faut bien comprendre la biologie, il faut bien comprendre comment ces thérapies-là fonctionnent. Il faut prendre le temps de bien les faire, bien les étudier. C'est un point très important, un point de sagesse, de ne pas perdre de vue l'importance de la connaissance, notamment de la biologie qui est en arrière de la thérapie. Yann, tu voulais rajouter?
Yeah, add something maybe to that. I would say also you need to understand the model, the new model of development, because you're completely changing things. Clinical trials are going to move at a much faster speed. Maybe the phase will be slightly different. The research and development process for the pharmaceutical industry, if you're starting to develop much more vaccine, it completely changes the blockbuster drug model that they use to make their revenue. So you have to think, if we're doing vaccine and we're charging much less money for those vaccines, well, where's the income then coming from for the companies? How's that going to affect the market and the access to these vaccines? So there are huge questions. So we need to accompany this research by actually social science research on the different models on all we're doing right now to see if we're proceeding in the right manner, and at the same time engage the population, communicate with people so that they understand what is happening, what is this new type of vaccine, what are the challenges, and be as transparent as possible in the process.
Vraiment des points. I'm going to go French. I need to translate also for the other. Tu veux-tu y aller, Yann? Vas-y.
Oui, ben je disais simplement que c'est important quand on fait une recherche scientifique qui est aussi un aspect autant transformateur que la recherche sur le RNA, qui va complètement changer la façon dont possiblement les essais cliniques se passent, dont on développe des médicaments, la façon dont l'industrie pharmaceutique construit son modèle de revenus. C'est des changements énormes dans la société qui impliquent beaucoup d'argent, qui impliquent un accès aux médicaments qui pourrait être complètement transformateur pour la population. Et c'est très positif, mais il faut que ça soit accompagné d'études en parallèle pour regarder quels sont les effets de tous ces changements-là de modèle sur la société, sur la façon dont l'industrie travaille. Est-ce que c'est sécuritaire? Donc, toutes ces grandes questions-là. Et en même temps, de la communication avec le grand public pour être très transparent sur où nous en sommes, qu'est-ce qui se développe, quels sont les risques-bénéfices que ça génère, pourquoi est-ce que c'est intéressant, etc.
Oui, et Yann Joly est directeur du Centre de génomique et politique, puis les considérations éthiques sont très importantes. J'ajouterais seulement que c'est comme ça qu'on veut développer la recherche sur l'ARN à McGill. C'est quelque chose qu'on veut sciemment accompagner d'une démarche au niveau de l'éthique aussi. So let's move to another question. Right? You guys are getting the jits here about switching French and English. You're probably getting a rhythm. I know most of them understand it. So let's move something a bit challenging. You ready? All right. When you present those new modalities, rightfully, people wonder, is this safe? And we're going to go and talk about the clinical trials that were raised. Jan, for example, mentioned that they're bound to accelerate, for example. So are the current modalities of RNA-based therapeutics subjected to the same clinical trials restrictions or profile as the other drugs right now? And I think Jan already mentioned the fact that it will accelerate, but in terms of safety, is it as rigorous right now for clinical trials with RNA modalities as the other drugs?
Can I just make a comment? I think there is commonly a misperception between emergency approval versus a full approval. And I think with mRNA vaccines in particular, because were in the middle of a pandemic, they were given emergency approval. And I do believe it was rightfully so, but of course the level of the safety data available was not what usually is required. But taking into account the circumstances, I think it was the right thing to do. When you are not in a constant emergency, so there is not like people dying around you, I think the level of scrutiny is going to be exactly the same. And I think it's, in the long run, it's probably going to be safer than the general drugs. And I would like to justify the statement because, as you mentioned, those drugs, both mRNA vaccines and oligonucleotides, are informational drugs, meaning that how they behave in the body and a lot of adverse events are shared. There will be unique biological sequence-specific and protein-specific things which will be different, but there is a lot of knowledge which is being shared and from one product to another. So I think because of this informational nature of this drug, eventually we will get to a much wider and very rich kind of understanding mechanistically what's going on, the safety profile, adverse events, and each additional approved drug will teach us more in terms of how to make it more efficacious and safer.
Mohamed
I would
Gabriel?
like to add a very important clarification, actually. Those regulators, actually, that looked at the files from Moderna and Pfizer during the pandemic, they did have some experience because there was clinical trial, phase 1, phase 2, for other mRNA vaccines that happened in the past. So people had some experience, right? And that's very important to know as well. They weren't less stringent, I would say. The EMA document was like 300 pages, right? And then there was a lot of scrutiny. They asked for a lot of data. And this data was provided actually after the phase 1 trial, phase 2 trials on the 50,000 people. Right? So I say it was emergency approved, but then we gathered a lot of data. Now these drugs or these vaccines, let me just talk about the mRNA vaccines. They are like a plug-and-play type of system, right? So you validated the LNPs, you know how it behaves. All what you're changing inside is the coding sequence. Even the signal peptides, for example, people don't want to change them. The UTR, they don't want to change them. So it's really the protein itself. So now I don't think also in the future that even though we have a large body of data about the carrier itself and its safety, the trials will continue being trials that are studying actually the effect of that protein and how your body is the efficacy and the safety of that protein itself. So trials will continue being as stringent, in my opinion.
All right. Je vais traduire rapidement. I will translate quickly. Really important points here. La question, c'est est-ce que les modalités à l'ARN, les thérapies à l'ARN ont été assujetties aux mêmes à la même rigueur d'évaluation pour les essais cliniques, par exemple. Dr. Korova faisait le point très important que pour le vaccin, par exemple, c'était au milieu d'une pandémie. Il y avait des gens qui mouraient, il n'y avait aucune solution ou à peu près pas. Donc, tout a été fait un petit peu en accéléré. Ça s'appelle une approbation qui était provisoire, on peut dire, temporaire, parce que c'était en temps d'urgence. Mais Mohamed Gabriel Alamé, qui est impliqué dans ces développements de ces technologies-là, dit que c'était vraiment bien documenté. Il y avait quand même une bonne substance de caractérisation clinique même pour les premiers vaccins, par exemple. Ce qui fait que c'était pressé, il y avait beaucoup de pression, mais il y a quand même une bonne rigueur. Ce que mentionnait aussi Anastasia, Dr. Korova, c'est qu'à la longue, pour certaines de ces modalités-là, le fait que ce soit des médicaments qui sont informationnels, c'est-à-dire qu'on change la séquence, mais que les bulles, les lipides nanoparticules, puis par exemple la composante chimique, l'ARN va être similaire, on va finir par accumuler des connaissances des effets secondaires de certaines de ces modalités-là, puis éventuellement on va savoir à quoi s'attendre pour un type de modalité. Il va y avoir des variantes qui viennent peut-être de la séquence de l'ARN, par exemple, en arrière, qui peuvent avoir des effets secondaires aussi, mais le gros de la modalité, on va savoir un petit peu à quoi s'attendre pour ce qui est comme effet secondaire. Je crois avoir capturé un petit peu. Yann?
Just maybe rapidly, just remind people, however, that there are different RNA therapeutics,
Yeah.
and that we're talking about the vaccine as we're saying that the modalities are now better known, and that there's a way that now this will be looked at much more quickly. But some of the other RNA therapeutics will require, will have an impact that will be more profound on the human bodies, and that will need to be documented and might require more of a case-by-case basis
Je
evaluation.
vais te laisser traduire, Yann.
Oui, je disais qu'il faut quand même rappeler, comme Thomas, tu nous l'as montré très bien au début, qu'il y a différentes choses qu'on peut faire avec l'ARN et que, bon, il y a les vaccins à ARN dont évidemment tout le monde connaît maintenant suite à COVID, mais et là, oui, une fois qu'on comprend le rôle de l'ARN dans ces vaccins-là et qu'on a analysé cette approche-là, l'approche reste sensiblement la même pour les autres types de vaccins qui vont fonctionner comme ça. Donc, on va pouvoir, si on veut, passer le stade de l'ARN plus rapidement au niveau de la revue clinique, et se concentrer plus sur l'aspect biologique du vaccin. Mais quand on va parler des autres modalités, des autres thérapies à l'ARN, dont certaines qui vont avoir un effet sur l'ARN sur le corps humain, et ou des fois par CRISPR, éventuellement, si on y arrive, à ce moment-là, on parle de thérapies beaucoup plus complexes. Et là, au niveau autant des essais cliniques et précliniques, il va y avoir beaucoup de travail à faire.
Merci.
If I may.
Yep, go ahead, John.
Science will win. That was our slogan during the pandemic. And really, I think that it speaks to the fact that safety is not imaginary. Safety is demonstrated through data, and it's demonstrated through clinical trials. And with our partners, BioNTech, we were able to provide a body of evidence, a body of data to regulators that showed that this was safe and that it was worthy of that emergency approval. There were no shortcuts made. Some of that acceleration actually came from at-risk investment, making sure that you're scaling up for those phase 3 clinical trials even before you see the initial data. So there's that aspect of speeding up manufacturing too. Every single decision point made by regulators and the drug developers, it really comes down to risk-benefit. And I think that we see emergency use authorization in certain pandemic scenarios. We see accelerated approvals quite frequently within the industry. And at each stage, the question is asked, is this safe and is it effective for this particular approval and this staged approval? And I think the critical thing to keep in mind here is that it's not that this is granted emergency use authorization and then it's done. Those vaccines have been granted full approval now, and it's still not done. You still have to do a post-market surveillance. You still have to understand and collect data to know that your product is safe. There's other formats for vaccines that are also interchangeable. And so there are licenses for your quadrivalent flu vaccine that is updated every single season, and it doesn't require additional clinical trials as long as it's within certain parameters and you're meeting those specifications. It'll be the same with messenger RNA going forward for their vaccines, and I think that really provides a benefit. But that doesn't mean that we take our eye off the ball and not look at safety. We always continue to monitor how safe and effective these products are, and we will continue to push for improvements on those products as we go.
Okay.
Can I just make
of course.
one comment in addition? I think there is no argument that the vaccines were transformative to our community, and there is no argument that they're safe and effective. When we're talking about the populational health, they're safe and effective. As with any drug, absolutely, you have a very rare adverse events. And I think right now, when we start to understand the biology of disease much better than we were understanding it previously, there might, sometimes it's also understanding what human characteristics associated with extremely rare adverse events. And I think the beauty of mRNA therapeutics that most likely they will be shared between all the future mRNA therapeutics, and if you have 0.001% of patients which reacted negatively, understanding what features or what status of those patients is related to those events. Can you comment on that, or do you feel, how do you think about it?
Can you summarize it into one question? You said a lot there.
How the informational nature of mRNA vaccines will allow them to become even more safer by identifying small subset of patients which might not benefit from receiving them.
Yeah, I think that if I understand the question correctly, you're really talking about the very rare events. And I think that when you have the broad vaccine campaign that we saw with billions of doses administered, I think that you are going to start seeing those very, very rare events. And then you need to figure them out and understand, okay, is this something product related, or is it something related to even the antigen? Is there something in the spike protein itself that is just a nature of the vaccine, but not necessarily the technology? And so can it be improved upon? I think that's some of the questions that we need to explore. But I think we also, you mentioned kind of unique cases, I think we also need to understand the durability of these vaccines and make sure that we're providing options to those that are most at risk. And so those that are immunocompromised, older patients that aren't able to seroconvert as easily. So I think that this is really important. I think in both cases you're talking about product improvements for the exception. And I think, yes, we should continue to move in that direction, and we are.
I'll make a translation pause here. So the question is essentially, la question, désolé, était est-ce que ces nouvelles modalités-là de thérapie à l'ARN étaient aussi sécuritaires que les autres pour ce qui est des essais cliniques. Puis, bon, la discussion continue autour de ça. Puis ce que John mentionnait, c'est que le devoir finalement de la compagnie, c'est de montrer que ce médicament-là, cette modalité-là, cette formulation-là est efficace, mais est aussi sécuritaire. Puis que cette responsabilité-là continuerait d'exister, mais qu'il y avait, dépendamment du type de modalité puis du type de maladie, probablement des critères un peu différents, mais que ça demeurait une évaluation qui était scientifique et très rigoureuse. Anastasia a apporté la Korova, Dr. Korova, c'était qu'il y a, à la longue, en utilisant les mêmes modalités de thérapie à l'ARN, on allait commencer à voir pour telle modalité, par exemple, quels sont les groupes de population, parce qu'on sait qu'il y a des groupes de population qui sont, pour lesquels le traitement ne fonctionne pas bien ou qu'il y a des effets secondaires, par exemple, plus forts. Puis en utilisant ces modalités-là, à grande échelle ou à la longue, on allait finalement être meilleur pour identifier ces événements-là puis orienter les bons traitements vers les bons patients, par exemple. Puis l'enjeu, c'était, pour ce qui est la réponse de John, c'était que si, pour des traitements, par exemple, comme le vaccin, il y avait des milliards de traitements qui avaient été distribués, on allait voir des événements très, très rares, par exemple des effets secondaires qui étaient à cette échelle-là arrivent rarement, mais qu'on allait pouvoir en soutirer de l'information puis identifier des groupes de population pour lesquels c'est pas souhaitable ou on allait pouvoir anticiper finalement peut-être une moins grande efficacité ou des effets secondaires supplémentaires. J'espère que j'ai bien traduit. So, Jan, you had another question or a comment about this?
Oui, juste rapidement. So rapidly, I just want to point out that this also emphasized the importance of pharmacovigilance after clinical trial. And that has been a bit of an issue in Canada during the pandemic because data access for pharmacovigilance was a bit of a struggle. And that's why providing broad access to data, especially when it's de-identified, in order for researchers, for pharmaceutical companies, to be able to identify the cause of these very rare events, to have enough data to be able to do that and not to miss anything, we're going to have to get much better at doing pharmacovigilance.
Je te laisse traduire.
Oui, ben je disais simplement que ça démontre l'importance aussi de la pharmacovigilance, qui est un stade après les études cliniques, une fois qu'on a commencé à administrer le vaccin à la population en grand nombre. Eh bien là, on doit être capable de mieux documenter les effets secondaires s'il y en a, de faire circuler l'information sur les effets secondaires pour qu'on puisse plus facilement faire de la recherche, identifier les causes de ces effets secondaires-là pour que le plus grand nombre de scientifiques possible puissent contribuer à cet effort-là. Parce que ça va être une étape, la pharmacovigilance, qui, avec des médicaments comme les médicaments avec l'ARN va prendre de plus en plus d'importance.
Merci. All right, so one last one in that drawer. How are secondary effects documented, and how transparent are they? Pharmaceuticals, for example, or clinical trials that are made, especially when it's quick to respond to a pandemic. How do they record it, and is it transparent?
By secondary effects, you're talking about some adverse
Yep,
effects?
adverse effects.
Yeah. You know, so part of the clinical trial, every clinical trial does have monitoring and reporting from those volunteers or from those patients that are enrolled into the trial. This is recorded stringently. There's a lot of publications as well. Obviously, there's filed documents with the regulators. But in terms of transparency, going back to the mRNA vaccines, there are a lot of publications on this in terms of the common adverse events. So it is available in the literature. I don't know if most of the public is actually accessing scientific literature, and that's maybe a different conversation to have. But there is information out there, and we try to be as transparent as possible.
Okay, thank you, John.
Yeah,
Mohamed Gabriel?
one thing actually, academically what happened during the pandemic is that there is a lot of sites all over the world that actually repeated certain of these studies with their own personnel. Like you can see, for example, doctors in hospitals in Italy or nurses or whatever, they were actually recording their own clinical data, if you want, and then they publish these. And these go hand in hand with whatever these companies have published as well. So providing more robustness to, and more, I would say, transparency. So.
I think transparency, I mean, we didn't reach complete transparency. You know, I always, as somebody that's interested in ethics and in public health, I think you always want more transparency. Yeah. I think that, yes, there is a reporting system. That's part of clinical trial. It's a robust reporting system where companies have to report these effects to governmental agencies. Are they now known to the broader public? Are they always published in the literature? The answer is no. But some company, that is a good approach to this. You'd want it to become part of the good practice that is done automatically by companies, and hopefully we're going to go towards that. But we haven't reached the point where we can say this is completely transparent yet.
I will double that I think in general, lack of transparency and reporting in clinical trial, not related to mRNA vaccines, but it's in general a practice. It's a problem. There was a lot of initiative coming within pharma, at least in US, trying to voluntarily force company to post data, both positive and negative, and it's a work in progress. And I would say with RNA, mRNA vaccines and RNA drugs, because they're informational drugs, so this positive and negative data from each clinical trial of this class can inform on a future clinical trial. So as much as people like you who are working on ethics can come up with ways how enhancement in this transparency and data sharing can be achieved, the better it's going to be. And I would feel Canada seems to be a little bit more normal than US. In US, everything right now, it's either black or it's white, and it's like everything is politicized, and clearly vaccines got heavily politicized. In reality, we should stay with the data, we should stay with the science, and transparency and open forum discussion is a way to eliminate this heavily, I'm trying to come up with a politically correct term. Maybe you can say it in French better, but you understand what I meant. Not like going to war about every single topic, but just keep it. Science will win, but to be able to educate the public, we need transparency.
Juste résumer rapidement. La question, je vais faire en français. La question, ça avait lien avec la transparence, par exemple, du processus pour rapporter les effets secondaires pour les traitements de l'ARN. John mentionnait que c'est très rigoureux, que des agences qui sont de façon très critique regardent les évidences, mais que, par exemple, c'est souvent publié dans des revues scientifiques, puis que c'est pas accessible généralement au grand public. Il y a eu plusieurs interventions par Yann et par Anastasia pour demander plus de transparence, pour mieux faire, pour rapporter ces effets secondaires-là. Anastasia mentionnait que les essais cliniques sur une modalité vont informer, par exemple, le prochain essai clinique. Ces choses-là, cette information n'est pas perdue, puis en transparence, en comptabilisant ces déclarations d'effets secondaires-là, on allait ultimement améliorer le processus. Puis il y avait un enjeu aussi dans la discussion de confiance, c'est-à-dire par rapport à la transparence, si on déclarait les effets secondaires à la longue, puis si on avait un bon processus plus transparent, de meilleure capture de ces déclarations-là, qu'en général, on allait probablement gagner le respect du public pour des nouvelles modalités, par exemple, qui peuvent être inquiétantes. Il y a eu un pitch en général, puis je croirais quand même un bon consensus pour davantage de transparence, puis d'accès à l'information au grand public de façon conjointe, mais de façon plus rigoureuse que ce que ça s'est fait dans le passé. All right, so I'll switch to the public questions. You guys are ready? So, all right, this past session were obvious ones, like major ones that everyone asked. These are actually from the public. I'll start with the one from Patrick. So, considering the properties of lipid nanoparticles, how can one think that they will remain to the injection site? So the question is, considering what's a lipid nanoparticle, does it stay there or is it distributed? Does it distribute all around?
So I'll answer that. So basically, animal studies and even human studies showed that these particles, particularly the ones that we're using now, they do not 100% actually stay in the muscle. So there is actually a kinetic where you have part of the dose that stays in the muscle. It makes a depot kind of effect, and then some of them actually leak out into the draining lymph node and into circulation. And that was also reported in the EMEA document. So it's not something that was surprising. We see that also in animal studies.
Do you think it can be responsible for secondary effects sometimes?
Some people believe so. We've done some studies, actually not in human, but in animals where we tweaked, actually, for example, either the size of the particle or the charge to keep the particles in the muscle. And then what you see is that a slight decrease in systemic cytokine circulations, but you can't ask a mice how do they feel, right? So that's not the problem. But I think it might play a role, but majorly I think the ionizable lipid is a very strong adjuvant, and it will kind of cause some adverse events.
OK, la prochaine question, c'est de Cheryl et Christina qui a été posée online. Is there hope for autoimmune disorders? Can they be treated by RNA technologies such as multiple sclerosis and so on and so forth? The autoimmune disorders. Anyone?
I can take that one. Yes, I sure hope so. I think that it is an unmet need, autoimmune disorders, particularly IBD, Crohn's disease, ulcerative colitis, but name them. There's many that impact our friends and families, including my own. So I think that we do have opportunities in RNA modalities for those disorders. The challenge there almost gets back to, I think, something that Anastasia had mentioned at the beginning of this, is understanding the disease. And I think when it comes to autoimmune disorders, it's really hard to come up with the right models preclinically to really optimize and target particular pathologies, because it's just very complex and it's very unique to humans. And it also can't be captured in necessarily just the petri dish, currently where we are with our understanding. But we'll get there. I think that these models are improving. There is more complexity to using human tissue and human organoids, and also using different models that hopefully will give us the opportunity to use that power of the RNA, and remember, the power of the RNA is that it's a genetic sequence. And so you can take advantage of the underlying biology, and you understand that biology, and now you can convert that and transform it into a medicine. But you have to understand the underlying biology first. But really some cool applications, I think, for autoimmune disorders could be delivery using the LNP to T cells, to B cells, to actually induce tolerance. That could be really nice for celiac disorder and other diseases like that. So I do have hope in this area. It's going to take a little bit longer, I think, than some of the other disease areas, but I do think that RNA can play a part.
Can I just make one more comment? The beauty of RNA therapeutics is informational drugs, and one of the aspects which have not been discussed is that the more clinical trials has been run, the more we de-risk the class-specific effects and understand how those drugs behave. This actually will create a true promise for autoimmune disorders, because right now, absolutely, you're right, the major issue, biology is complex and it cannot be modeled in animals pretty much robustly and reliably. So what I'm alluding to is that when we start to better understand the classes of modalities and how they behave, it's quite possible that we might be able to run clinical trials and get those drugs checked in humans as quickly as possible, and this will get us to the cure faster. Okay. But of course, ethical considerations of that is, it will keep you employed for quite a while.
Yeah, Mohamed Gabriel?
Yeah, I also have an addition to that topic. I think at least in animals, we can see a large body of evidence now that you can induce tolerance with these mRNA therapies, especially messenger RNA ones. But there's a very important point when we want to address these diseases. These are based on inflammatory diseases, right? It's very, very highly inflammatory. So we need to take into consideration also the delivery system. You need to have a delivery system that's immune silent, like the mRNA inside of it. And a lot of work from BioNTech now is using certain liposomes, and a lot of people, including us, we're developing these formulations that do not actually induce high cytokines. And these will be, I think, the future to induce tolerance. So there is also a component of the delivery system that we need to think about.
All right, I'll translate a little bit here. There were two questions. Il y avait deux questions que je dois traduire. La première était en lien avec les lipides, les nanoparticules lipidiques, c'est le terme en français. La question était de Patrick qui posait la question, considérant que c'est un complexe qui peut se redistribuer dans l'organisme, on peut pas s'attendre, comment on peut s'attendre à ce que ça reste au site original? Puis Mohamed Gabriel mentionnait que non, ça reste pas, c'est connu que ça circule. La question sous-jacente, par après que j'ai posée, c'est est-ce que ça peut être responsable pour des effets secondaires? Mohamed Gabriel disait possiblement, mais c'est pas à ce point-là clair qu'est-ce qui est responsable de ça. Il croit que c'est plus la composition en tant que telle de la composante. Puis à la fin, il disait que pour éviter ces effets secondaires-là, on va les améliorer. Il y a moyen de changer les composantes des lipides à la surface pour que ça reste, par exemple, vraiment au site d'injection puis diminuer les effets secondaires. La deuxième question qui était très importante, je crois, motiver tout le monde, c'était la question sur des maladies auto-immunes. Est-ce que les thérapies à l'ARN et ces technologies à l'ARN pouvaient être utilisées pour des nouvelles thérapies sur ces maladies-là qui sont très diverses, par exemple comme les scléroses multiples? La réponse à ça, John mentionnait qu'il croit que oui, mais la difficulté souvent, c'est que c'est difficile d'avoir un modèle animal qui reflète vraiment la nature de cette maladie-là chez l'humain. C'est dur. Il croit vraiment qu'éventuellement, on va développer ces choses-là, mais c'est des choses assez complexes, puis il n'y a pas vraiment de bon modèle pour ces choses-là pour l'instant. Anastasia mentionnait que c'était la clé, encore une fois, c'était vraiment la nature informative ou informationnelle de ces thérapies-là qui permettent vraiment de mieux les développer, de mieux les cibler, puis qu'éventuellement, on allait avoir un meilleur arsenal plus précis pour des thérapies comme celle-ci. Puis, Mohamed Gabriel, veux-tu traduire ton point? J'en ai un petit peu.
Moi, en fait, Ces maladies, normalement, ils ont une composante très immunitaire, basically. Et il y a beaucoup de cytokines qui sont impliquées et de l'inflammation qui est impliquée. Et les systèmes de livraison actuels qu'on utilise pour les vaccins à ARN sont très inflammatoires. Right? Donc, si on veut vraiment induire la tolérance, ça c'est une stratégie de faire, il faut développer des systèmes de livraison qui ne sont pas immunogéniques. Et c'est un effort que BioNTech fait. Nous, à UPenn, on est en train de travailler. Et puis, il y a plein d'autres compagnies qui essayent de changer soit les formulations ou même de découvrir des lipides qui sont moins immunogéniques pour induire la tolérance. Et dans des études précliniques qui ont été publiées il n'y a pas longtemps, il y a des formulations, en fait, qui fonctionnent. Et, at least dans les modèles animaux, ils montrent une induction de tolérance qui est très acceptable. Donc,
un français impeccable.
Merci beaucoup.
Une question de Mélanie. Question from Melanie, I'll translate later on. Can these new discoveries lead to treatment against long COVID and other infections that have an effect in the long run? Persistent infections. We see a lot of them, and there's quite a few people who are affected by the long COVID, for example. Any taker?
Yeah, I don't know. I think we're still trying to understand. We, as in the community, collectively are trying to understand long COVID and some of the symptoms there and the underlying pathology, how they relate to the initial infection, and the genetic component that likely plays a role in that too. So I don't know, and I think that there is a lot of work that's going to be put into understanding that better. And again, once we understand those mechanisms, then we can start to develop drugs around them.
That's good. Okay, so the answer to this is we don't know. There's a lot of people working on this, and again, one major en français, sorry. La réponse à cette question-là, c'est on ne le sait pas encore. Il y a beaucoup de gens qui étudient le phénomène de long COVID, mais on ne comprend pas encore les fondations biologiques de la COVID longue, par exemple. C'est quelque chose qui est revenu, je n'ai pas traduit aussi efficacement que je l'aurais dû. Régulièrement, nos panélistes mentionnent que la clé, c'est vraiment de comprendre la biologie, puis de ne pas perdre de vue les fondements biologiques pour chacun des traitements, des modalités. On est excité par toutes ces thérapies-là, mais il ne faut pas vraiment perdre de vue ce qui se passe au niveau mécanistique à l'arrière de ça. Donc, l'importance de la recherche sans trop mettre les bœufs avant la charrue. La charrue avant les bœufs, c'est le contraire. Oui, merci. All right. Une question one question from Gurpreet. Can these medicines and how can be personalized for every individual?
Le concept de vaccin est un concept qui est difficile à personnaliser, parce que bon, on veut
en anglais en premier, comme ça on va rester dans le même processus.
Yes, sure.
Dans le même problème depuis moi.
The concept of vaccine usually speaks to the collectivity, or to a certain amount of people in order to achieve the complete immunity of a population. So it's actually really interesting when we speak about personalizing vaccine. So I think there are aspects of the vaccine that could be personalized, but in the end, it will become vaccine that will be developed for masses, or then we're speaking to personalized medicine, which is a bit of a different concept. So it also depends on what you're trying to achieve there.
Yeah, maybe we could just expand and go right into the the cancer vaccine applications of this, and using a patient's what you call a neoantigen, and providing that as a vaccine to that patient so that you can activate the immune system so that the immune system recognizes the tumor. This is an approach that several companies are pursuing, some with messenger RNA, other ones with actually peptides or other types of modalities, but the idea is the same, that you are able to profile that patient, understand what the genetic mutations are in that patient. You then have a computational aspect of this, and this is where I think that the critical part that is still being developed is you need to be able to transfer those mutations into epitopes or antigens and hope, or at least have evidence to suggest or hypothesize that those mutations are going to lead to presentation of antigens on the surface of the tumor. And that's a really key part to this, is that they have to be exposed on the surface of the tumor so that your immune cells can actually identify that tumor and then cause a cytotoxic effect. And so I think there's other aspects to that too. Certain tumors are what you call hotter than others. They're more immunosensitive than others that are colder. Melanoma is an area where this is actually really working well. But solid tumors, breast cancer, ovarian cancer, it's a little bit more challenging because there are immunosuppressive microenvironments in those tissues that even if you actually have the vaccination that's working, your antigens are presented on the tumor, your immune cells are ready to go, they go towards the tumor, and then they're shut off, essentially. There's some defense mechanism that the tumor has essentially evolved to have. So I think that this is a really cool concept, and it's one of the things that actually enabled the very rapid generation of the messenger RNA for the COVID-19 vaccine, because there was already a process in place that would allow something like a 3 or 4 week turnaround time. When you're dealing with cancer patients, you want to do this very rapidly, right? And so you have you have a lot of work that needs to happen from needle to needle, as they call it. So you're going to take a liquid biopsy, you're going to take a blood sample, you're going to take a sample of the tumor, then that's going to go to the lab, they're going to convert that, they're going to do all the computational work, they're going to make a messenger RNA, LNP, send that back in, and then that's treated with the patient. You need to do this fast if you're a critical cancer patient. So I think that this is an enabling technology that did help for the COVID-19 vaccine, and is also maturing in its own application, and we're starting to see that in melanoma and other areas too. You can take the same approach and actually make it more generalized, so you don't need to have the neoantigens every time, and you can have up to 20 neoantigens, I think I've seen. But certainly having that personalization does provide a unique way to address individual cancers.
Je vais traduire rapidement, à moins que Mohamed Gabriel, tu étais sur le bord, là.
Non, ça va.
Okay. So, la question c'était, est-ce que ces médecines-là, basées sur les fondements de l'ARN, est-ce qu'ils peuvent être inclus comme des médecines de précision personnalisées? Ces deux concepts dont on entend parler, les médecines de précision, médecine personnalisée, donner le bon traitement au bon patient, pas surtraiter, puis tenir compte finalement de la condition soit génétique ou environnementale, ou même culturelle, ou populationnelle d'un individu. Ces médecines de précision. Yann a commencé par dire que pour certains vaccins, non, c'était pas quelque chose qui allait être vu comme des vaccins, par exemple, pour des infections ou pour beaucoup d'applications. Ça va demeurer une médecine précise, mais ça ne sera pas au niveau de l'individu. Mais il y a d'autres modalités, il y a d'autres cas, comme John mentionnait, par exemple, que dans certaines formes de cancer, il y a une médecine très, très précise qui peut s'exercer, qui est basée sur les principes de néoantigènes. Pour des cancers comme le mélanome, par exemple, les cancers du pancréas, il y a des essais cliniques en cours présentement où ce qu'on fait, c'est on séquence l'ARN ou le génome des tumeurs du patient. Puis on identifie des petits bouts qui sont uniques à la tumeur qu'on appelle néoantigènes. Il y a des essais cliniques comme ça où rapidement on a l'information qui est unique à la tumeur puis qui est absente de la partie en santé du patient. Puis on peut générer des thérapies sur ces fondements-là. C'est une médecine qui est tellement précise qu'elle s'exerce dans un patient en particulier. Puis les mêmes principes-là sont en train d'être développés par beaucoup de groupes puis qui vont éventuellement probablement pouvoir même s'appliquer sans que ce soit vraiment à un individu parce qu'il va avoir suffisamment de données accumulées pour qu'il puisse dériver finalement des pratiques de cette base-là. Donc la réponse, c'est pas pour toutes les applications. Des fois, c'est plus la population, mais pour d'autres applications comme les médecines de précision ou personnalisées en cancer, par exemple, c'est en développement et c'est excessivement précis. OK. I have a question that I think is very important, and I've heard multiple patients talk about it. The question by MMC was whether there were specific protocols developed for patients, rare disease or autoimmune disease, that were not making it traditionally in clinical trials. You need to have sometimes large groups of people to make it in clinical trials. Will RNA-based therapeutics allow patients that were ignored by the traditional clinical trials to actually get in finally and have access to innovation?
So I can comment on oligonucleotide-based therapeutics and the mechanism which does exist in US. It's N01 and it's a compassionate trial. And the difference is essentially is a goal of the regulatory agency like FDA is to ensure that crazy scientists do not cause harm. And this mechanism should exist. And they also want to ensure that there is no negative impact on public health. So whenever you start to talk about ultra-orphan indications, there is no public health. There is only informed consent. Because we're talking about one physician, one drug, one patient. I'm exaggerating, okay? One physician, one drug, 3 patients. And in this case, the informed consent is essentially, we need to ensure that the patient understand the consequences, what will be done, and it's truly the informed consent. In terms of how trials are, like changes in how our trials are being designed right now, what is commonly being practiced is within a patient dose acceleration. Because normally when you run the clinical trials, you start with a very low dose, which is never can be efficacious by any stretch of imagination, but you have to show that it's safe. You do it in a cohort of patients, and it's a different cohort of patients where we are getting towards efficacious dose. So right now, FDA already allowed to do within the same patient dose acceleration, and this is a common way how patients finally can get access to this truly disease-modifying drugs, but in a safe and responsible fashion.
If you want to know how things are in Canada, because I think we need to differentiate that, and for better or worse from what happens in the US, is you put it at slow speed, quite slow, too slow when it comes to precision medicine. A regulatory framework is really underdeveloped for the potential of precision medicine. So things take more time to get approved, and as far as exceptional access processes, they're also much more complicated for patient to have access to these medication on those terms, and the N-of-1 trials are much more complicated to set up as well. So there's a lot of work from a regulatory perspective that our policymakers at Health Canada and at the provincial services also have to do for us to catch up.
Actually, what FDA, usually nobody is saying good things about FDA, but what they did, they recently came up with a recommendation for ultra-orphan, like N-of-1 or N of several indications, is simplifying the clinical development process, preclinical development process. Because if you try to make a drug, the major cost is GMP manufacturing and IND-enabling tox studies in non-human primates. And the cost of that per drug, even if you do everything super efficiently, is around $3 million. So when we're talking about academic PI developing a drug for one patient, access to this type of funding is close to impossible. Or maybe you can do it for one crowdsourcing, but we need to have a systematic approaches. And essentially what FDA has altered their demand on type of IND-enabling tox studies which need to be in place for specifically this ultra-orphan indications, and that you can proceed safely with very small doses and then accelerating within a patient, but without doing this multi-million-dollar studies in non-human primates, which often are not even predictive.
We need to innovate more in terms of how the regulatory process, just as we can innovate in science, we have to also be able to innovate at that level, because otherwise you hit a roadblock at some point, and then you see things continue to develop in other countries and not in your own country, right, where you started the process, which can be very frustrating. So that's definitely something that remains to be done in Canada.
Frustrating for people developing treatments, but
frustrating
imagine
for
the patient,
everybody.
The patient.
Yes, exactly.
Yeah, for sure. So, en français, la question qui était vraiment excellente de l'audience, c'est dans le cas de maladies rares, rares où il y a très peu de patients affectés, où il y a une concentration locale assez limitée, souvent ces patients-là sont exclus des essais cliniques. Il n'y a pas assez de monde, c'est pas stratégiquement avantageux pour développer ces thérapies-là, où il y a différentes raisons au niveau légal ou au niveau du processus gouvernemental pour développer ces essais cliniques-là. La question, c'est comment est-ce que les thérapies à l'ARN peuvent permettre à des patients qui étaient ignorés par le mode traditionnel d'essai clinique à finalement avoir accès aux thérapies. Dr. Korova mentionnait par exemple qu'il y avait aux États-Unis des modes, des façons avec pas vraiment un essai clinique dans le même sens du terme, mais des essais, on appelle ça ultra-orphelins, où un petit groupe de patients est mis d'accord avec un médecin, avec un traitement particulier qui est particulièrement prometteur, puis que c'était vraiment dans ce petit groupe-là, là, dans une modalité qui était vraiment locale, à petite échelle, qui pouvait être testée comme ça. Dans le cas, par exemple, de patients qui souffrent puis qui n'ont pas beaucoup d'espérance de vie, bien, ça fait beaucoup de sens. Il n'y a pas beaucoup d'autres options. Hein. Puis on n'a pas le temps d'attendre que les essais cliniques se passent. Yann mentionnait que souvent, ce qui est frustrant dans les modes traditionnels, c'est pas juste l'accès, mais c'est aussi la lenteur du processus puis de la politique en arrière qui fait que c'est très, très long au niveau gouvernemental d'approuver l'accès à ces traitements-là. Donc, il y a vraiment un enjeu là, au niveau de l'amélioration à la fois du mode pour traiter ces patients-là à petite échelle, mais aussi au niveau de l'accélérer puis améliorer le processus. Puis Yann mentionnait le fait qu'on doit innover là comme on innove en sciences, par exemple. All right. We're nearing the end of the panel. I would like to ask you one about CRISPR. I picked it. Is that okay? We didn't talk about CRISPR yet. Okay. Some clinical trials are underway that explore the power of CRISPR genome engineering, considering that this form of therapy can lead to changes in the genome, in contrast to other RNA therapeutic modalities who just come in, get degraded, do their job and get degraded. This one actually changes the genome. Can they be presented or promoted to the public on the same safety grounds as the other RNA therapies, such as a vaccine?
No, I mean, they're different, definitely. So that doesn't mean that they're all bad or that we should never do any of this. I think, first of all, we have to differentiate between germline therapy in an embryo and somatic therapy. Germline therapy being something that you do that is going to affect future generation. So it must be really considered extremely carefully before we decide to go into human, if we were to go there, and only for the most serious disease. I think that's the consensus at this point in time. But in the case of somatic therapy, there's already some clinical trials, and there's already, I think, one or two, and my colleagues are probably better at counting those successful therapies, so more, so that are already there. So there's definitely great potential, although these are expensive, very expensive therapies. So there's probably something to think about in terms of equity of access to those therapies, and how we can ameliorate that from that standpoint as well.
Can I just make one comment? I think for somatic genomic editing is not that different. It's a gene therapy. It's permanent solution, but it's once and gone. So I think what is happening right now is that RNA therapeutics, which we're discussing today, going head by head with a gene therapy, including CRISPR. And we have seen the actual life example happening. With oligonucleotide therapeutics or mRNA vaccine, you get temporal, long-term, but temporal modulation of gene expression. And when you clinically show for many years that affecting this gene in this patient population with RNA drugs is safe, then it paves the way for actually CRISPR therapeutics come and provide a permanent cure. I think it would be very hard for CRISPR to go in right away, and we with RNA drugs are paving the ways. The true life example is TTR therapies, where oligonucleotides and siRNA showed right now it's a transformative treatment for people with this disease. And right now, as an Intellia came up with a CRISPR, which looks absolutely fascinating. It's one example, and it's done and gone. what they're going to charge for it, it's a different question. But on the technology front, it's an mRNA with a couple of oligos in this lipid bubble. So it's not complicated.
Yeah, I just want to add where I see CRISPR being a viable option to be, because I totally agree with you that it should come after validating using oligo, for example, right? But where I see it, there's certain diseases where the newborn dies immediately after birth, right? So you can do in utero gene therapy, and that's where you can basically, but it's complicated, I know, but that's where I see that they can test it as first.
I think ethical complications of that is going to be tremendous, because you cannot guarantee that it's not going to change the genome for something which would be inherited. I personally believe somatic gene editing is probably following de-risking by RNA therapeutics is more realistic, at least
So it
short
can
term.
still be somatic. But anyway.
On a un petit début de débat ici. We have a beginning of a debate here. I will translate if that's okay, John. Okay. La question c'était essentiellement les technologies d'édition génomique basées sur CRISPR-Cas9. Ces technologies-là sont guidées par l'ARN, mais mènent à un changement au niveau de l'ADN. Il y a un enjeu là, parce qu'on ne change pas de façon temporaire, mais de façon permanente le génome, puis ça a un impact, des considérations éthiques assez importantes. La question, c'est est-ce qu'on peut traiter ces thérapies-là comme des thérapies à l'ARN, comme d'autres modalités? Il y a vraiment eu de belles réponses ici. Il y en a commencé par dire que non, simplement non, c'est pas les mêmes enjeux, mais qu'il y a une différence quand même au niveau de la sécurité, au niveau éthique, entre une édition somatique dans des cellules, par exemple, qui vivent et qui meurent. Par exemple, tu fais une édition, ça guérit la maladie, puis ça va être remplacé ultimement, ou tu vas mourir avec, alors que l'édition germinale, la lignée germinale, est transmise selon les générations. Puis là, c'est vraiment des considérations éthiques profondes. Si on parle de l'espèce ici, c'est des éditions de l'humaine, de l'espèce, il y a vraiment un enjeu majeur. Anastasia mentionnait que pour l'édition somatique, ce n'est pas un enjeu majeur parce que tu mets des ARN avec une bulle de nanoparticules lipidiques, puis que tu guéris la maladie dans le soma, ça ne change rien. Mais qu'il y a vraiment un monde de différence entre ça et une édition germinale, par exemple, au niveau éthique. Elle suggérait aussi que les thérapies ASO ou siRNA qui étaient temporaires allaient précéder puis servir de preuve de principe pour les thérapies à édition génomique, par exemple. On a utilisé d'autres modalités d'ARN pour montrer qu'on peut traiter la maladie, puis une fois que ça fonctionne bien, on sait que en traitant cette cible-là, on arrivait à une conclusion avantageuse. On allait possiblement passer au niveau de l'édition génomique, puis entre-temps gagner sur la modalité de livraison aussi. Puis il y a eu un enjeu ici avec Mohamed Gibrel qui disait que s'il y avait beaucoup de potentiel pour des traitements dans ces cas-là pour des maladies qui étaient autrement pas possibles, comme des cas, par exemple, de mort in utero, des différentes éditions somatiques qui permettaient à l'enfant de vivre, là, par exemple, ça avait un potentiel important. Puis Anastasia mentionnait, le danger ici, c'est qu'on peut traiter la lignée somatique dans in utero, mais c'est très difficile de garantir qu'il n'y aura pas une édition germinale aussi, qui serait transmise selon les générations. J'espère que j'ai bien capturé la conversation. Thanks. I got a couple of...
Aussi en français, le coût de ces thérapies-là pour l'instant est très dispendieux, est inaccessible pour la grande majorité de la population de la planète. Donc, il faut aussi en être conscient. Ce n'est pas un enjeu scientifique, mais c'est un enjeu au niveau de l'accès et de l'équité au traitement, de ne pas accélérer la différence entre les riches et les pauvres à travers les traitements qu'on développe.
L'accès au traitement. We're at 8:32. John, if you're burning to add something, you're good. All right, folks, we had really an extensive discussion. I think we had a great contribution by each of our panelists. Please join me to thank them for the discussion. On vient de remercier les panélistes pour l'audition ce soir qui viennent clore. On vous remercie pour votre participation. Thank you everyone at home for listening to us, and we'll see you around, probably around RNA at some point, soon or far. And keep an eye on these folks. They're really innovators, and they also keep an eye on the considerations such as ethics, for example. They're really great thinkers. So we're grateful to have you here tonight. as well. Thank you.